And so next up is Mary Ghering from MIT, and she's going to talk about her work in the plant's syncytium, or maybe even a little later in development, and her work in using single-cell nuclei to get at that single-cell nucleus. Go ahead, Mary. Thank you.
Thanks, Ken. And thank you for the invitation to speak here today and for all of you for joining me. So I'm going to tell you about the application of single nucleus RNA-seq to endosperm development. And the endosperm is a very fascinating tissue that is an essential part of seeds. So if we look here at an Arabidopsis seeds, there are 3 genetically distinct components: the seed coat, which is formed from maternal integument tissue, and inside the 2 products of fertilization, the embryo, which is diploid, and the endosperm, which is triploid, because it has 2 maternal and 1 paternal genome. The embryo and endosperm are genetically identical, except for this addition of an extra maternal genome in the endosperm, but have very different fates. So the endosperm is ephemeral. It doesn't transmit any genetic material to the next generation, but it's absolutely essential for the development of viable seeds. So it's known in many interspecies crosses, or even crosses within the same species, that hybrid incompatibilities can arise due to defects in endosperm development. One of its primary functions is to transfer nutrients from the maternal parent into the developing seed, and then the embryo will assimilate those nutrients as it matures, or later as the seedling germinates. Endosperm is also really important to us because the majority of the calories that we eat come from endosperm, primarily in the form of the staple grains. Endosperm is a very interesting tissue because it is this biparental tissue. It's a product of fertilization. It has maternal and paternal genomes, and its function is to serve as a nutrient sink for the maternal parent. And so because the endosperm is biparental and it's not perfectly related to the tissue from which it's extracting nutrients, it's thought that there may be genetic conflicts within the endosperm itself over this process. And so one idea is that because the endosperm has maternal and paternal genomes, those 2 genomes may have different optimal expression levels of genes that are involved in the nutrient transfer from the maternal tissue into the seed. And because of that, that potential genetic conflict, we think this epigenetic phenomenon called gene imprinting evolved. So imprinting describes a situation where alleles of genes are expressed differently depending on whether they're inherited from the male or from the female parent. And this occurs in flowering plants and in placental mammals, but in plants occurs only in the endosperm tissue. So let me just describe to you in this cartoon form what imprinting means. So most genes in the genome in a given tissue, we would expect they would be expressed biallelically, meaning from both the maternally and paternally inherited copy, or they might not be expressed at all. Imprinted genes are different. So MEGs are genes that are expressed only from the maternally inherited copy, and PEGs are genes that are expressed only from the paternally inherited copy. And we know that some of these imprinted genes, when they're disrupted, they do cause defects in endosperm and seed development. In terms of how imprinting is controlled, we know that differences in epigenetic modifications between maternally and paternally inherited genomes in the endosperm are important for this differential expression. So the endosperm genome in Arabidopsis and in many of the other species is DNA hypomethylated, so it has lower DNA methylation due to active demethylation that occurs before fertilization. And in Arabidopsis, we know that a lot of this demethylation occurs at transposable element fragment sequences. Additionally, there's also differences in chromatin modifications between the maternal and paternal genomes, such as increase in the polycomb mark H3K27 trimethylation on the maternal genome in some regions compared to the paternal genome. These differences in epigenetic state between the two genomes are closely tied to the mechanisms of imprinting. For example, I'll focus on paternally expressed imprinted genes for most of this talk, or PEGs. Many of the PEGs that have been identified are associated with transposable element fragments that are differentially methylated between maternal and paternal alleles, with the methylated allele being the expressed allele. And the silenced maternal allele is associated with H3K27 trimethylation. So my lab and many others have been looking at imprinting on a genome-wide scale since around 2010, both at identifying imprinted genes and then looking at epigenetic modifications associated with imprinted genes. And so the way we identify imprinted genes is like this. So we'll take two strains of Arabidopsis, say Columbia and LER, do reciprocal crosses, and isolate F1 endosperm a few days after fertilization. So this is a hybrid endosperm. We perform RNA-seq, and then for those sequencing reads that overlap SNPs between, let's say, Columbia and Landsberg, we can assign them as coming from, as being transcribed from the maternally inherited allele or from the paternally inherited allele. And so for every gene in the genome that has SNPs between the two ecotypes, we can determine the fraction of transcripts that come from the maternal allele. So what you see at a genome-wide scale is that this looks like a pretty normal distribution centered around 67% maternal for most genes. So for most genes, they're expressed in the same dosage that you find the DNA, this 2-to-1 maternal to paternal ratio. But what we're interested in are these imprinted genes over here at the edges of the distribution. So there's a few hundred imprinted genes with usually more MEGs identified than PEGs in a given species. The sorts of genes that are imprinted are variable. So there's not a lot of Go enrichment categories for MEGs. We see lots of different types of genes being imprinted. For PEGs, there are enrichment for transcription factors and chromatin modifiers. So one feature that we noticed a long time ago from doing these types of global RNA-seq analysis was that often you have genes that are very biased. So this gene here is 80% of the transcript from the single paternal allele and 20% from the two maternal alleles. But they're not quite like that cartoon I showed you where there's complete monoallelic expression, only paternal expression, no maternal expression. And so I've wondered for some time, what does this partial imprinting mean? So it could be that both alleles, maternal and paternal alleles, are expressed in endosperm cells, but at different levels. So that here you still have more paternal allele expression, but it's not completely monoallelic. Or it could be that there's heterogeneity in imprinted expression among endosperm cells, so that you do have completely monoallelic expression in these 3 cells, but not in this 4th cell, so that when you do an RNA-seq, you get this partial imprinting result. A third possibility is that there's technical issues confusing the matter. For example, if there's contamination of maternal seed coat and genes are expressed in both the endosperm and seed coat, that could make everything appear more maternal than it is. So we set out to try and distinguish among these possibilities. And I think the answer could be important for both understanding the function of imprinting and also its mechanism. So if we find, for example, that there's variation in imprinted expression among endosperm cells, that might also suggest there's variation in the epigenetic programs that control imprinting. And so we would then need to figure out how those are established and maintained in different endosperm cells. So to do this, we took a single nucleus approach. So basically, we decided to do single nuclei RNA-seq to both see if we could learn more about transcriptional programs and cell types in the developing endosperm, and also to determine whether imprinting is heterogeneous in the endosperm. So what I'm going to tell you about today is posted in a manuscript on bioRxiv,
and was spearheaded by an excellent former graduate student in the lab, Colette Picard, with contributions from 2 postdocs, Becky Puvalis and Ben Williams. Okay, so one of the reasons that we did nuclear RNA-seq, of course, there's a lot less RNA in a nucleus than in a whole cell, but the reason we needed to do this is because the endosperm is syncytial in its early development. So there's actually no cells to do protoplasting on in early endosperm. Rather, there's what's called nuclear cytoplasmic domains organized around the endosperm, and then cellularization happens at about the heart stage of embryogenesis. So we took whole seeds, primarily at 4 days after pollination. We isolated nuclei, and then stained the nuclei with DAPI. What we can take advantage of to enrich for endosperm nuclei is this difference in ploidy. So the endosperm is triploid, but the embryo and seed coat are diploid. So then when you sort the nuclei, you can sort individual nuclei primarily from the 3C and the 6C peaks. Although, of course, we get some bleed over from these other nearby peaks, particularly the 4C peak. So we sorted individual nuclei into wells. We decided to do a SMART-seq2 prep for the library prep. And then we sequenced about 1 million or 2 million reads per nucleus from about 1,500 nuclei and detected around 4,000 genes per nucleus on average. So pretty good coverage. We do know something about domains within the endosperm. We know it's not just a homogeneous bag of nuclei, there is structure there. So this is a confocal slice through the endosperm, through the seed. Here's the embryo at a late globular stage, and then this is the endosperm here. In this seed, the endosperm has started to cellularize. So there are 3 known endosperm domains. The micropolar endosperm surrounds the embryo, and we know it's important for signaling to the embryo, developmental patterning of the embryo epidermis. The peripheral endosperm is involved in nutrient storage and mobilization. And the callosal endosperm down here interfaces with the maternal tissue. And unlike the rest of the endosperm, where cellularization of the syncytial endosperm proceeds from this pole here to this pole, the callosal endosperm remains free nuclear for much longer. So we did this on reciprocal crosses between Cole and CVI, sequenced about 1,500 nuclei, and were able to enrich for about 1,000 endosperm nuclei. We performed clustering on the endosperm nuclei and also on the seed coat nuclei, but I won't go into that. And we did this separately for the reciprocal crosses. So we have Cole female crossed to CVI male and CVI female crossed to Cole male, F1 endosperm. We found that we liked this program SC3, this unsupervised clustering, which uses several different techniques and basically tells you how often 2 nuclei cluster together using these different methods. So what you can see is we got between either 14 or 11 clusters. Some of these are are sort of tighter than others. So you can see here, this largest cluster, these nuclei are similar to one another, but certainly not as tight as this cluster here. And so our clusters range in size from 4 nuclei to about 90 nuclei. So much as Bert was telling you, we want to understand, okay, what are these clusters? Do they actually represent different nuclei types or cell types in some cases? So we took advantage of a very nice data set that Bob Goldberg and John Harada's lab had published several years ago, where they did laser capture microdissection on the 3 different domains in the endosperm, and that was published in this paper in 2013. And so from that data, which was microarray data, you can assign a set of markers for peripheral, micropolar, and callosal endosperm, and then look to see, so then we look to see what is their expression of those markers across the clusters that we defined. So when you're looking at these plots,
for those of you that aren't familiar, the size of the circle is the fraction of nuclei that a gene was detected in. So the bigger the circle, the more fraction of nuclei in that cluster was detected in. And the color is the average expression level. So what you can see is that we do have some very nice, this cluster 6 is very enriched, for example, for callosal markers. But we have some clusters that don't have really any enrichment for known markers. And we have other situations where, for example, for callosal endosperm, we find 2 clusters, clusters 12 and 13 in this cross, and clusters 1 and 6 in the other cross. So we then performed differential expression analysis among all possible combinations of the clusters. And so we could identify genes specific to each cluster and do things like go term analysis on those. So we wanted to understand if some of the clusters we identified represented physically distinct regions of the endosperm. So we chose to do this by in situ hybridization. I'll just show you the results for 2 marker genes. Again, we were interested in this finding that it looked like there were multiple callosal endosperm clusters in our data. So this gene, AT5G10440, came out as specific to one of those clusters, cluster 12, and cluster 1 in the reciprocal cross. And when we performed in situ on it, we could see it here in the callosal endosperm, but not throughout the entire callosal endosperm region, just in these nuclear cytoplasmic domains lining the cavity here. So anatomically, these are known as callosal nodules. This other marker was also in putative callosal endosperm clusters, but it was enriched in the other one, so cluster 13 and cluster 6. And by in situ, you can see that this is now in the most basal portion of the callosal endosperm in what is known as the cyst. And I really love this in situ because you can see these nodules actually sitting on top of the cyst with no expression, no RNA detected from the marker compared to down here, even though they're physically touching. So what we've defined from these data is that the callosal endosperm, which has been thought of as this one region, actually has distinct nuclei types. And so then you can perform Go analysis on these different clusters, for example. And whereas they're all enriched for this function killing cells of other organisms, which was actually already known from the Belmonte paper about callosal endosperm, we can see that the nodules, for example, are specifically enriched for folate biosynthesis, and the cyst very nicely is enriched for phloem sucrose loading, which is really cool because this is the part of the endosperm that abuts the maternal seed coat tissue. Okay. And so through many of the same approaches that Birch has described, we were able to identify new types within these 3 known endosperm domains. I haven't been able to go into all of the details of these different clusters, but some of these seem to have, some of these clusters are actually due to cell cycle phase, so S and M phase really stand out. Some are due to different functions. Okay, so let's get back to the question of imprinting now that we've defined these different cell types or nuclei types. So there are a lot of challenges for looking at imprinting in individual nuclei. So now we're really only looking at a subset of those transcripts that have SNPs. And so some of the challenges are, of course, there's lots of zeros in the data. And when there's a 0, is that because we just didn't detect it, or because actually the maternal allele was silent and the paternal allele was expressed, for example? Transcriptional bursting kinetics, if they're different between the maternal and paternal alleles, may also lead to sort of false positive calls for imprinting. And so we decided ultimately that it was going to be almost impossible to say something about an individual nucleus. Like I can't say, okay, in nucleus number 85, this gene is a PEG. Rather, we still need to rely on groups of nuclei. And so what we did is across those 14 or 11 clusters we defined, we looked at imprinting in those clusters to see whether or not there was variation in imprinting among the different types that we had defined. So first we can see, first we look at just where are imprinted genes expressed in the endosperm. Is there any variation among nucleotypes? I didn't put in the slide for the MEGs, but essentially the MEGs are not enriched in any particular domain of the endosperm. They seem to be expressed throughout the micropilar, callosal, and peripheral endosperm. But the PEGs clearly had enrichment in callosal endosperm. So this is, we looked at enrichment of expression of the PEGs in the different clusters that we defined. And what you can see is that about half of the PEGs are enriched for expression in the callosal endosperm. Some are specific to the nodules here, some higher in the cysts, and some high in both the cyst and the nodules of the callosal endosperm. So what does this higher expression of PEGs represent? Again, coming back to our cartoon of an imprinted gene where the paternal allele is expressed and the maternal allele is not expressed. Higher expression could mean that there was a loss of imprinting, so that now both maternal and paternal alleles are contributing to expression. Or it could be because there's increased paternal allele expression, so there's even more paternal bias. So from our data, we were also able to look not at just total gene expression, but allele-specific expression, obviously. And so now if we calculate a maternal-paternal enrichment score, if something is more blue, it's become more paternal compared to the other clusters. And if something is purple, it has become more maternal. And what you can see is in the bottom right corner, again, we have blue. So it looks like the higher expression that we see, or the enrichment of PEG expression that we see in callosal endosperm, is due to increased expression of the paternal allele. So greater paternal bias in the callosal region. I'll just show you one specific example of this for this gene that fell into this category. So here, Colette has drawn across the different clusters the maternal counts in red, the paternal counts in blue, and then just the simulated 2x maternal counts, because again, remember there's that difference in ploidy between maternal and paternal genomes. So what you can see is that in the nodules and the cyst of the callosal endosperm, this gene is expressed from the paternal allele, no counts from the maternal allele. And this we're looking at here in total, more than 100 nuclei. Whereas in this cluster 5, you can see that the gene, you get a bump in expression from the maternal allele, lower expression from the paternal allele. So the gene really doesn't look imprinted in this cluster. What we're interested in now, of course, is trying to understand, well, how do you actually generate these sorts of expression patterns? I don't have the answer for you. So we're thinking about how cluster-specific imprinting might be generated. It could be that there's a distinct epigenetic state in the callosal endosperm, and we have found when we look at the expression of epigenetic regulators that there are a set that are specifically expressed in that part of the endosperm and not in other parts. Some of those are themselves PEGs. It could be that we're talking about the paternal allele being upregulated, that there's callosal endosperm specific transcription factors that interact differentially with maternal and paternal alleles, perhaps due to a difference in epigenetic state between those alleles. And so those are the questions that we're exploring for the future. Okay, so I'll end there and summarize. From doing this fairly small study from endosperm at 4 days after pollination, we've been able to uncover transcriptional complexity that we didn't know about in the endosperm, including that within known domains, there's additional complexity. We find that There's little variation in MEG expression or in their extent of maternal bias across different endosperm nuclei types, but that in the clausal endosperm, there's the highest and most paternally biased expression of PEGs. And again, this is really interesting to us, getting back to the idea I introduced at the beginning of conflict in the endosperm between maternal and paternal genomes, that you have highest expression of PEGs at this region of the endosperm that interfaces with a maternal parent. And so there may be the paternal genome may be trying to exert some influence over that nutrient transfer. In the future, we'd really like to now expand, not look at just transcription, but at things like DNA methylation at the single cell level to try and pair these data. We're looking to see if there are transcription factors specifically expressed in the clausal endosperm that might explain this upregulation of the paternal genome that we see. And ultimately, we'd like to extend this throughout seed development. We've just looked at one stage here, and in multiple species as well. So again, I'll end by acknowledging Colette, Becky, and Ben, who did really fabulous work here. We've been supported by the excellent core facilities at the Whitehead Institute and MIT. We've gotten advice on single cell approaches from other labs at Whitehead, and funding from the NIH and NSF. Thank you.